cell organelle, and we are going to discuss the most important organelle of the cell, that is the nucleus. Nucleus is the largest component of the cell. It is a double membrane bound by dense protoplasmic body and controls the cellular metabolism. It encloses all the genetic information and is able to transmit the same to the next generation. Robert Brown coined the term nucleus. J. Hammerling, he was working with a unicellular green alga, marine alga, that is the acetibularia, proved that nucleus regulates the activity of cytoplasm and also the heredity. Except mature mammalian RBC, sieve tube or sieve cells of the phloem, every living cells have a nucleus. Prokaryotes have an incipient nucleus, that is, devoid of nuclear membrane, nucleolus and histone proteins, and such type of nucleus is referred as nucleoid, or you can call it as chromatin material. You can see the nucleoid and the distinct nucleus. Nucleus usually occupies 20% of the cell mass. Its size depends upon ploidy of the nucleus, cell maturity and cell function. Nucleus lies near the geometric center of the cell, and peripheral nucleus present in adipocytes and plant cells. It is rounded in young plants and polygonal in animal cells, oval or elliptical in mature plant cells. Karyotheca, or the nuclear membrane, the outermost covering of the nucleus, also called as nucleolemma. Term was given by Erclap and Hertwig. Karyotheca composed of two unit membrane, and they are separated by a distance of 150 to 300 angstrom. White perinuclear space. Endoplasmic reticulum is connected to the outer nuclear membrane at several places. Nuclear membrane is perforated by nuclear pore of the size of 400 to 1000 angstrom diameter with a space of 1500 angstrom. Nuclear pores are covered by an octagonal structure composed of 8 granular subunits called as annulus or annuli, or you can call it as bleb. These pores regulate transport between nucleoplasm and cytoplasm. Small ions like potassium, sodium, and chloride move through nuclear membrane, while tRNA, r-RNA, and mRNA pass through the nuclear pores. Protein nucleoplasmin, which occur in pore complex, regulates the nucleocytoplasmic traffic. Now here you can see the nuclear pore, the perinuclear space, the nuclear envelope, inner and outer membrane. Nucleoplasm or karyoplasm or karyolymph or nuclear sac, all this is named for the same thing. Nucleoplasm contains high concentration of nucleotides which occur in the form of triphosphates. Nucleoplasm have enzyme for glycolysis. Thus, nucleus obtains its own energy for glycolytic pathway. This is the nucleoplasm. Inside nucleoplasm, there is a network of fine threads. These threads are called chromatin fibers. Chromatin threads composed of DNA, RNA, histone protein, and non-histone protein. Chromatic threads contain genetic information, and chromatin network appears differentiated into two parts. You can see here Euchromatin, light-stained diffused region. This part have less histone and more acidic protein. Euchromatin is more active part of chromatin, and its DNA undergoes rapid transcription. You can say it is genetically more active. During the nuclear division, Euchromatin replicates in S phase of the cell cycle. It takes part in crossing over. It is formed by loose spiralization of the nucleosome strands. Heterochromatin, dark stained, well delimited, thick and condensed part of chromatin. This part has more histone and less acidic protein. Heterochromatin is genetically a less active part of chromatin. Heterochromatin form stop points in transcription. Now you can see the dense area is heterochromatin, transcriptionally silent. Then you have euchromatin, which is transcriptionally competent. It is formed by solenoid type of coiling of nucleosome containing DNA strand. It does not take part in crossing over. Replication occurs late in S or G2 phase. Chemical composition of chromatin. Chromatin threads composed of DNA, which account for about 31%, RNA 2 to 0.5%, and proteins, that is histone part is 36% and non-histone part is 28%. Histone and proteamines are basic proteins. Histone protein composed of mainly arginine, lysine and histidine amino acids. Proteamines have more arginine, but lysine is absent. Histone occupy the major groups of DNA at an angle of 30 degrees to the helix, and it neutralizes negative charges of internucleotide phosphate in DNA. In prokaryotes, polyamines have similar functions. On the basis of relative amount of arginine and lysine, there are 5 types of histone proteins, that is H2A, H2B, H3, H4 and H1. H3 and H4 are arginine rich, H2A and H2B are slightly lysine rich, and H1 is highly lysine rich. Non-histone protein of chromatin mainly composed of tryptophan and tyrosine amino acids. Under electron microscope, chromatin appears beaded. Beads are called nucleosome or new body by Woodcock and Ploidy in 1975. Nucleosomes are 11 nanometers in diameter and 5.7 nanometers in height. Nucleosome model of chromatin proposed by R. Kornberg, and each nucleosome consists of 140 pairs of nucleotides and 8 histone molecules. Now here you can see the nucleosome connected with a linker DNA and H1 histone protein. Central core of the nucleosome composed of histone and nucleotides of DNA form 2 coils, that is 1% around this octamer of histone. 2 molecules of each, that is H2A 2 part, H2B 2 part, H3 2 part, H4 2 part. So they are present. The core particle of the nucleosome is in the form of octamer. Nucleosomes are linked in a string by thin threads called linker DNA, and linker DNA composed of 60 pairs of nucleotide and 1 histone molecule, that is H1. Nucleolus, discovered by Fontana and name was given by Bowman. It is a naked structure that is attached to chromatin at a specific point called as nucleolar organizer region, or in short, you can call it as NOR. NOR is a type of heterochromatin. There is normally one nucleolus with each set of chromosomes. True nucleolus is also termed as plasmosome, and they are stainable with acid dyes. Cell actively engaged in protein synthesis have a large nucleolus. Nucleolus contain the following parts. Fibrous part, that is the central part of the nucleolus, composed of fibers called nucleonema. Nucleonema mainly composed of rDNA and proteins. Granular part, peripheral part of the nucleolus, composed of granules. It has r-RNA plus proteins, which represent the maturing ribosome phase. Amorphous matrix or pars amorpha, a proteinaceous matrix which contains both fibers and granules. r-RNA, that is 5.8S, 18S, and 28S, are synthesized in Nucleolus. Ribosomal protein is synthesized in cytoplasm. Nucleolus also play important role in the initiation of cell division. And the nucleolus